All right, welcome back. It's Miss Roman again. This is part 3 of our review session for the Unit 7 test, which covers chapters 12 and 13 from your textbook. In the last 2 videos, we talked about mitosis, meiosis, how the stages are similar, how the stages are different, what you need to look for when you're looking at a picture of a cell to determine what stage of the cell cycle it's in. And this last video, we're going to focus our attention on linked genes and the law of independent assortment and the law of segregation. Linked genes are genes that are found on the same chromosome. And we study them because they are inherited as a group because they're on the same chromosome. So we have 46 chromosomes, 23 from mom and 23 from dad. And we are diploid because we have these two sets. The chromosomes for humans are numbered 1 to 23, and you have one set of 23 chromosomes from your mom and one set of 23 chromosomes from your dad. They're numbered 1 to 23, with chromosome number 1 being the longest of the 23 chromosomes, and then they get smaller. So chromosome 17 would be smaller than chromosome number 5. And you see here a homologous pair. The one chromosome from dad in blue and the chromosome from mom in pink. And then you see they have marked the gene A, and this could be for eye color, attached earlobes, cystic fibrosis gene, all kinds of anything. But A is the gene. It codes for a trait. And there are different versions of that gene. So if this was eye color, the gene would be eye color, but then your version of the gene, it could be blue eyes, green eyes, brown eyes. Of course, it is way more complicated than that, but we're just using this as an example. So let's say this is for eye color, and big A was the allele for brown eyes and little a was the allele for blue eyes. Then the two versions of those alleles on those chromosomes, we would call that the homologous pair. And then whatever color eyes you have will depend on the inheritance pattern for that gene, which we will learn in the next unit. When it's time for your cell to divide, or if this is a homologous pair in a germ cell, and germ cells undergo meiosis to make your gametes, then if this is a homologous pair in a germ cell, when the cell enters into meiosis, once it gets the signal to enter into meiosis, the cell will move from G1 into the S phase. There will be a replication of all of the DNA. So during the S phase, you begin to have duplicated chromosomes because when the second copy is made of that chromosome, they stick together through their centromeres. And then we call those identical sister chromatids. But because they're still connected at the centromere, they are considered one chromosome together. And then we can also call these the duplicated homologs. Now, of course, your chromosomes can have thousands of different genes on them. And if you look on the internet and look up the Human Genome Project, they have mapped over the years more and more chromosomes that are on our 23 chromosomes. And so a particular chromosome could have the gene for the sodium-potassium pump, whether or not your second toe was longer than your first toe, if you have attached earlobes or not, how tall you are. All of your traits are spread out over those 23 chromosomes. So any genes that are on the same chromosome are called linked genes because they're on the same chromosome, and then they would be inherited as a group. So whatever's on chromosome number 15, you would inherit that chromosome as a unit, so those traits would be inherited together. So that's why sometimes family members will have a lot of traits in common. It might be because they were on one chromosome together. So any sibling that got that chromosome had all of those traits together. And when genes are linked, the way that we can determine if genes are linked and then how close together they are on a chromosome, you can begin to determine the distance between two genes on a chromosome by looking at the gametes themselves. Because remember, the gametes will be haploid, and they'll only have one copy of each duplicated chromosome and only one of the homologous pair. So all of the gametes will have the sister chromatids that have been separated into the daughter chromosomes. And by looking at the unduplicated haploid chromosomes in the gametes, you can determine if crossing over has occurred. And then if it has occurred, how far away are genes that are on these linked chromosomes? So the pictures you see here, actually, I will try to make this bigger. Hopefully it'll work. Okay, I may or may not have actually made that larger. But if we look here, we have a homologous pair where genes A and B are very close to each other, and then below we see a chromosome pair where genes A and B are very far apart from each other. And when the tetrad forms during prophase 1, crossing over can occur because the chromosomes are in such close proximity. And if genes are very, very far apart, then the chances of crossing over happening are very, very high because there's just a lot of space for crossing over to occur. So if we looked at the gametes for this homologous pair, we would expect to see a lot of gametes that have the results of crossing over. However, if the genes are very, very close together, crossing over occurring in between those genes is really not that likely, purely based on the fact that there just isn't many places that crossing over can occur in between those two genes. So if we looked at the gametes, we would expect to find little a with little b most of the time, and big A with big B most of the time. And then very few gametes would have little a and big B together, and few would have big A and little b together. Whereas in our other homologous pair here on the bottom, finding big A and big B together in a gamete on one chromosome is probably just as likely as finding big A and little b on a chromosome together inside of a gamete. And these images that also come from oops, that also come from the Khan Academy. Let's see, I'm gonna make this a little bit smaller. Sorry, there we go. Obviously, I was born in the 70s and older. Okay. These images from the Khan Academy show you the possible gametes and the chances of finding a gamete with this combination of genes. If the genes are linked and close together, we don't expect a lot of crossing over to occur between the genes. So if we looked at all of the gametes, we would have a very low percentage of gametes that had these combination of alleles, just because the probability of crossing over occurring is low. However, if we look at genes that are far apart on a chromosome, if we took the gametes and looked at them, we would have a pretty equal probability of finding any combination, either little a with little b or little a with big B. The chances, there's a much higher chance of seeing crossing over, and it's purely because they're so far apart. There's just a lot of places that crossing over can occur. So it makes it more probable and more frequent. If we look at genes that are not linked, so they're on different chromosomes, then how they sort into the gametes is random. Little a could sort into a gamete with big B, or it could sort into a gamete with little b. It's not a result of crossing over that these gametes have the alleles big A, little b. Whereas the gamete here on the right that has a big A and a little b is due to crossing over. So even though if we just look at the alleles, they look like, they're both big A, little b, which they are. But the way that those alleles got into the gametes are different. On the right, this gamete received a little a allele and a little b allele because of crossing over. And the gamete here on the left that has a big A allele and a little b allele, it got its alleles just because of random sorting of the chromosomes. This is not a result of crossing over. But if you were just looking at the alleles present in the gametes, you would not know if that was a result of crossing over or if it was a result of the independent assortment of the chromosomes into the gametes. Now, if you're told that these are linked genes, then you can determine how far apart the alleles are from each other or the genes are from each other by looking at how likely that combination is. So how likely is it that we would have these gametes? And if we were told that these were linked genes on the right, on the bottom right, and the bigger the probability, the more likely it is to occur, the farther apart the genes are. The smaller the probability of having this gamete means that the likelihood is very, very low, which means that crossing over is not frequent, and therefore the genes must be close on the chromosome together. All right. Last thing we need to talk about is the law of independent assortment and the law of segregation. Now, you already know the law of independent assortment and the law of segregation. You just don't know that you know it. The law of independent assortment describes something that you already know. We just haven't given it a name. And the law of segregation describes a process you already know, but now we're just putting a name to it. The way that our original cell, our original germ cell, divides and becomes the gametes, you know how to describe what's happening between each cell. And you know that during the G1 stage, certain events happen. During the S stage, certain events happen. During the G2, certain events happen. And then each stage of meiosis 1 and meiosis 2, you know what to look for. You know how to describe them. And when G1 gets the signal to go into the S phase, we call that DNA replication during the S phase. We make a second copy of the DNA. It has a term, it has a phrase that we call it. So when we say DNA replication, we know it's the S phase. We know that means a second copy of the DNA is produced. So it's similar for the law of independent assortment and law of segregation. We're just giving a process its own name. And students get really confused when you add in the names because it can throw you off because you think you're learning something new, but you're not. You're just putting a name to a process you've already learned. And the law of independent assortment and the law of segregation describe the chromosomes during meiosis 1. So if we look at this image from the Khan Academy, of course, and we look at our G2 cell that is going into meiosis 1. Remember that in prophase 1, the tetrads form. So these are actually the tetrads here, all right? As they go through prophase 1 and then end up on the metaphase plate in metaphase 1, what happens as the cell is moving from prophase 1 to metaphase 1 is, the law of independent assortment. So let's see if I can, oh my goodness gracious. All right, technical difficulties. That's what we'll do. Okay, so this is the law of independent assortment. Let's, okay. The law of independent assortment happens right here. The result of the law of independent assortment is what metaphase 1 looks like. Metaphase 1 shows that the law of independent assortment has occurred. The law of independent assortment says that when the tetrads line up on the metaphase plate, they have no idea what any other tetrad is doing. It is completely random. So having both of the blue chromosomes on one side and the pink on the other is just as likely as having one pink and one blue on one side of the metaphase plate and the blue then pink on the other side. The law of independent assortment states that the homologous pairs sort independently into the gametes. And where it actually occurs is, you can think of it as pro-metaphase 1. If the law of independent assortment did not happen, then none of these cells on the right would be possible. Only the ones on the left. If you were trying to prove to someone that the law of independent assortment did occur, and they didn't believe you, if you showed them this cell or any of these cells over here would prove that the law of independent assortment happens, because every single one of these cells has one blue and one pink chromosome. So the law of independent assortment had to have happened. The cells on the left don't disprove the law of independent assortment because this is a possibility. It just doesn't prove the law of independent assortment. If you were trying to prove to someone the law of independent assortment absolutely happens, you would want to show them one of these cells, because there's no doubt that these are the result of the law of independent assortment. These could be, but they don't prove that it can happen. Okay, this is a possibility, but this, beyond the shadow of a doubt, it occurs. Now, the law of segregation occurs here between metaphase 1, during anaphase. During anaphase 1 is when the law of segregation occurs. And the law of segregation states that the homologous pairs segregate. They move away from each other. The tetrad is broken, synapsis is broken, homologous pairs are separated. We have a diploid cell here. Now they are haploid. The law of segregation explains haploid cells resulting from anaphase 1. If you wanted to prove that the law of segregation occurs, then you would want to prove that the homologous pairs are segregated. They are separated. They are in completely different cells. They're not a homologous pair anymore. Any of these 4 cells, which would be telophase 1, and everything after, prove the law of segregation, even if it's 2 blues together, because these are not homologous chromosomes. They're different chromosomes, and they're 1 each of different chromosomes. That's what the law of segregation says. The law of segregation says that you will end up with haploid cells, no matter if they're all blue or all pink. You just have 1 each. So if you want to prove that the law of segregation occurs, any of the cells from telophase 1 to the very end prove that law of segregation occurs. You wouldn't use metaphase 1 to prove the law of segregation because at this point they are still tetrads and it's still diploid. Prophase 1, still diploid. We can't prove law of segregation. It could happen in the future, but that cell does not prove law of segregation. Does occur. Lining up on the metaphase plate in metaphase 1 doesn't prove law of segregation. It's still very much diploid. Anaphase 1, we know that telophase comes next, so it would be better to say that telophase 1 is the first indication that the law of segregation is valid. It is true. It does happen. Anaphase 1, you could get very technical, but that would probably be better described as the process. Like if we wanted to see the law of segregation in action, we would watch anaphase 1. We could justify how we got to the law of segregation by looking at anaphase 1. And then same thing for law of independent assortment. If we wanted to show the mechanism, okay, fine, we have these cells that have different combinations of the homologous pairs are lining up differently. But what caused it? You would see it during prometaphase. Prophase, they are not moving to the poles at all yet. So prometaphase would be the correct answer of how would you show that it is actually happening? Like, how does it get there? Prometaphase for law of independent assortment, and then prometaphase 1 for law of independent assortment, and then anaphase 1 for law of segregation is what you would use to prove that law of segregation is occurring. Now, some review questions for you. If this cell was on the test and I asked you, what is the chromosome number for this cell? What would you say? I would say the chromosome number is 8. There are 8 chromosomes. 1, 2, 3, 4, 5, 6, 7, 8. 8 chromosomes. So chromosome number is 8. Are there homologous pairs in this cell? Yes, there are homologous pairs. We look for length to determine homologous pairs. So we have 2 long, 2 medium long, 2 medium, and 2 short. So those are homologous pairs. So is this cell haploid or diploid? The cell would be diploid because there are homologous pairs. Those go together. There are 2 of the long, 2 of the medium long, 2 of the medium, 2 of the short. So that makes it diploid. If we had 3 longs, 3 medium long, 3 medium, 3 short, it would be triploid. Are these chromosomes duplicated or unduplicated? This cell, the chromosomes are unduplicated. We don't have sister chromatids, so that means that these chromosomes are not duplicated. All right, let's look at this cell. What's the chromosome number for this cell? There are 1, 2, 3, 4, 5 chromosomes, so the chromosome number would be 5. Is this chromosome haploid or diploid? There's an odd number of chromosomes, so it has to be haploid. There's no way to have homologous pairs, and that would make it diploid. We can't have homologous pairs because it's an odd number. And I forgot to ask you, for our cell right here, we said it's diploid. So what is N equal to if this is diploid? N would equal to 4 because a diploid cell, we would say 2N is equal to the chromosome number, which is 8. So if 2N equals 8, then N is 4. And we do have 4 pink chromosomes as one set and 4 blue chromosomes as another set. This cell, because it's haploid, N is equal to the chromosome number, and there are 5 chromosomes, so N is equal to 5. All right. What about this chromosome? What is the chromosome number? All right, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Okay, I counted 19 chromosomes. So the chromosome number would be 19. Is it diploid or haploid? It has to be haploid because there's an odd number. And that means N is also equal to 19. These chromosomes are not duplicated. There are no sister chromatids. All right, this cell, what is the chromosome number? The chromosome number is 6 because there's 1, 2, 3, 4, 5, 6. Is this cell diploid or haploid? This cell is diploid because it has homologous pairs. There are 2 long, 2 medium, and 2 short chromosomes. So it's diploid. There are homologous pairs. If it's diploid, 2n is equal to the chromosome number. So 2n is equal to 6. That means n is equal to 3. And we do have 3 pink and 3 green chromosomes. So one set is 3. Now these chromosomes, are they duplicated or unduplicated? These are duplicated because we have the identical sister chromatids attached at the centromere. All right, our last cell. What is the chromosome number for this cell? It's 5. 1, 2, 3, 4, 5. So the chromosome number is 5. Is it haploid or diploid? It has to be haploid because there's an odd number. There cannot be homologous pairs if there's an odd number of chromosomes. So this is haploid. There are no homologous pairs. So n is equal to the chromosome number for haploid cells. So n is equal to 6. Are the chromosomes duplicated or unduplicated? They are duplicated. We have 2 sister chromatids attached at the centromere. So notice that the cells that have the duplicated chromosomes have 6 chromosomes. The sister chromatids do not count as a chromosome because they are connected at the centromere. When these cells go through anaphase and the sister chromatids are pulled apart, at that time they now become chromosomes. So this cell cannot be in G1 because it has duplicated chromosomes. That only happens in the S phase. So this is either the end of the S phase, G2. This could be prophase. It cannot be metaphase because they're not lined up at the plate. Cannot be anaphase because they're duplicated. And if this cell were to be in anaphase, we would have 10 chromosomes in that cell. Because once the sister chromatids are separated from each other, they are now considered chromosomes. So these kinds of questions: how many chromosomes, diploid or haploid, duplicated, unduplicated, what is n equal to? There will be questions like that on the test. All right, if you have any questions, just email me and let me know.